Last segment. So this segment is going to be mainly about your nucleic acids. And the difference here in terms of molecules, lots of carbon, lots of hydrogen, lots of oxygen, lots of nitrogen. And this time we have a new player called phosphorus. And phosphorus comes into play in something called phosphate. What we have is a phosphate group. That's what you hear the monomer called in a second. I'll show you. So we have a PO4 group. That's our phosphate. That's where the phosphorus comes into play. And we're going to talk about phosphates and their role in different things in the energy molecule. And then we're going to talk about how the energy molecule is related to DNA. And you're just going to be so thrilled the rest of this year. So let's talk about the monomer for nucleic acids. The monomer for nucleic acids is called a nucleotide. And your basic definition here is that it's a sugar, a phosphate, and a nitrogenous or nitrogen base. So I do have a couple of pictures up at the top. We have two sugars. If we have ribose as the sugar, then we have ribonucleic acid, also known as RNA. Ribo coming from ribose. If we have deoxyribose, then we have deoxyribonucleic acid, or DNA. So we don't really have a polymer of these guys because the polymer is the nucleic acid itself. We don't have an intermediate, really. I guess you could say ribonucleic acid, or RNAs, have the different forms, and so therefore that could make different polymers if you want. But there really isn't a way to describe it. Like you can't have a di-deoxyribonucleic acid. That doesn't exist. You can't have, a polyribonucleic bond. Like, those things don't exist when it comes to your nucleic acids, which is why you see it, not on your list a lot for your summary chart and things like that. So I want to take a closer look at the sugar and the bases on their own. So I'm going to give them their own slides by themselves, but I wanted to show you what they look like together and just kind of sum up how the nucleotides are going to be put together. So let me do that for you. Okay, so the nucleic acids are named after their sugars. If you didn't pick that up in the last frame. And by quick glance, you would probably want to say, hey, those look like isomers. And I would say, hey, you're onto something, except for they are not going to have the same chemical formula. In ribose, we have an alcohol group present on, and this is going to become important. Let's go back and think about how we talked about numbering carbons in a cyclic. So we're starting at 12 up here, going to the first carbon that comes about clockwise. So this would be carbon 1. This is carbon 2, 3, 4, 5. So these are pentose sugars. They are monosaccharides. And on carbon 2, in ribose, there is an OH group. However, on carbon 2, in deoxyribose, we seem to have lost the oxygen. D, meaning without, to get rid of. If you decontaminate something, think about what you're doing. Deoxy. We got rid of the oxygen. There's just a hydrogen. So the sugars are similar, but not exactly the same. And it's important to make sure that you can tell whether or not you're looking at a ribonucleic nucleotide or a DNA nucleotide. So now let's take a look at their bases. I wish I had a bigger picture like you have in your notes, but the other ones are way too big to put into the video, and this one is just too small. But we have two types of nitrogen bases. They get their name nitrogen base or nitrogenous bases because there's nitrogen in the ring. So that's how they get their names. Notice that the rings here, yeah, they have carbons in them. There's a carbon here, there's a carbon here, there's a carbon here, carbon over here, and a carbon over here. But there's also nitrogen embedded in those rings. So that's how they get their nitrogenous base name. But we have two different groupings. We have the purines, which, as you can see, are double ring structures. In other words, there's two rings connected to each other. And then we have the purines, which are all single rings. And you may know them better by just their letters. You may have learned something about A, G, C, U, and T. And you may have learned A always pairs up to T, and G always pairs up to C in DNA. When we're working with RNA, A is going to match to U. T is going to match to A. G is going to match to C, and C is going to match to G. And this is when we're going from DNA, which are these guys, to a transcribed RNA. So when we put together the DNA strands, there are always a purine to a pyrimidine. So essentially, that means there are always three rings across. So let's take a look now at what the nucleotides, when they are all pieced together, their phosphate, their sugar, and their nitrogen base, actually appear in strands like. Okay, I tried to find the picture that you have in your notes. I try to find the best match to them every time that I am looking for them, but I could not find one that would work within the program for the video. So we're going to have to make do with this one. Not my favorite one, but it serves our purpose. So I want to talk about this information here where it talks about the 5' end and the 3' end. So, okay, jump back a couple of steps. Let's talk about DNA versus RNA. We already know the sugars are different. We already know that we have U in RNA, but T in DNA. So the last thing that makes the two different, oops, hello, how about a 3? Oh my goodness. There we go. Is that RNA is single-stranded and DNA is double-stranded. SS single-stranded, DS double-stranded. And you should get used to that because you see them written like that, because we can also have single-stranded DNA, but let's not talk about that right now. So let's go back to talking about this 5' end, 3' end deal here. I want you to notice that we give them direction. This is called strand direction. So we can say we're at the 5' end of a strand, or we can say we're moving towards the 3' end of a strand. And that's how we denote the direction of the strand. And what I want you to really pay attention to is that they're named by the carbon that is at that end of the strand. So here's our sugar molecules in here. Here's our first carbon, our second carbon, our third carbon, right? 1, 2, 3, 4, and there's our 5th carbon. That's how this gets to be the 5' end. Down here, 1, 2, 3. Here's carbon 3, so that becomes 3' end. We also talk about DNA being said to be complementary. The strands are said to be complementary. Essentially, it means they match each other. However, they run opposite directions. That's part of the match. That's part of them being complementary to each other. So you can see the matching side to the 5' end on the other strand is the 3' end, and the matching side to the 3' end is the 5' end. So on this strand, the nucleotides are literally flipped upside down, if there's actually a upside to or a correct orientation to a nucleotide. I think we make the assumption there is. So it's literally upside down. So those are some major differences between the molecules themselves. And again, just wanted you to get an idea of the organics in terms of the sugar, in terms of the phosphate group, in terms of what a nitrogen base looks like, and the directional because of the sugar, carbons. That's really what we want to accomplish here now in terms of DNA versus RNA. And the first, portion of the DNA chapter is going to sound very familiar to this, just to refresh everybody's memory that we did talk about this already. But, in general, that's basically what I wanted you to have a look at. There's one last thing I want you to see. I want you to remember talking about the going from DNA to RNA to protein that we talked about in the last couple of segments of the protein section. But for now, there's only really one more thing I want you to see, and we're going to see if we can get the picture to cooperate in terms of DNA. So let's flip to that picture. So this is a crazy picture. Again, yours is in black and white anyway, but it's definitely not the red and white and blue one. If you look at the notes in the electronic file, that picture in color would be red, white, and blue, I think it is, or something like that. But this is kind of cool, yellow and purple. I'm game. Purple being one of my favorite colors, of course. So these are different, what we call forms of DNA. And you always have all three forms present. And there's purposes to all three forms. But I wanted you just to be aware of the fact that you have these. Two of them are right-hand twists. And one is a left-hand twist. And we'll get into talking about why there's a left-handed twist and why are there two forms that are right-handed, A versus B. But I just want you to take a couple of minutes to notice the differences. You have these spaces in here that are very large. Big space in here, called the major groove. And then you have these spaces that are over here called minor grooves. Okay, over here, minor grooves. Okay. What I want you to understand is that they all serve a purpose. There is a purpose for those major groove spaces being that exposed. That's a lot of exposure there. And that's important because that exposed area is going to be key to the start of a gene, to an important piece of information for something. There is a reason why the DNA is exposed like that. And I just wanted you to see that there are different forms of the DNA. They're not always perfect double helixes like you see in a lot of the pictures. When DNA is actually coiled up inside the cell, inside the nucleus, you get these different forms. And that's really the last thing I have to say about your nucleic acids for now. Again, they get an entire chapter to themselves, so there's not much more we need to kind of throw around here. So essentially, my dears, you are done with the chapter at this moment. So where you move to from here is up to you.